So all of these things that I've discussed so far target the bacteria directly, much in the same way as traditional antibiotics target the bacteria directly. But many people believe that targeting the host, influencing the host immune response, will have a benefit as well. Now, it could have a benefit, and it could be very broad spectrum benefits. But what many people with those concepts in mind fail to realize that when you target the host, you're likely to increase side effects. The animal models show variable potency. I mean, they look like they're active, these approaches, but just not as good as traditional antibiotics, so we don't know how they'll work in humans. We don't know how to predict which ones will work in humans. You have to take polymorphism of the human target into account. What happens in immunocompromised patients, which is the majority of patients with the infection, we don't know. We don't know whether animal models are suitable. We don't know if we'll have to migrate to non-human primates, or if you're modifying the human immune response, will even non-human primates give you any sense of what will happen in humans? And all of these things increase the cost and the risk of the program. So it's quite likely that no biotech or pharma is going to take on developing that type of new product on their own. It's possible it will have to be done in a public-private partnership, or perhaps entirely with public money to demonstrate that this very innovative approach works. So a key question is, how much would that cost and how long would it take? So we can analyze that. There's almost no chance that this type of host defense or innate immunity will deliver a drug by 2025. And if you project plan what it would take to get a project to registration, so that's going 2 projects at phase 3, 4 projects at phase 2, 8 projects through phase 1, 34 preclinical projects, it costs around £600 million. And that seems like an outrageous figure. I said that to somebody in pharma, and they said, well, if you could guarantee we could register a project for £600 million, we'd bite your hand off. So it's not a massive amount of money. It's a very realistic number to get there, okay? But that's not going to happen easily, right? So if we want to find out if host defense altering the way humans respond to the infection is the way of the future, we have to work out how that's going to happen. So given the current alternatives to antibiotics portfolio, we cannot expect a new therapeutic to treat systemic or invasive bacterial infection within the next 10 years. Okay? So that's what the analysis tells you. Now, since then, Aradis has shown some interesting information. Maybe they'll squeeze it in. But we could also look at this and slice it a different way, that say, with adequate funding into antibodies, probiotics, and vaccines, we can expect 1 new medicine for C. difficile by 2019, and we've seen an antibody has appeared. 1 for Pseudomonas, it will be an antibody or a vaccine by 21, and 1 for Staph, an antibody or a vaccine by 2022. So it's not terrible. There will be new approaches, not very many of them, most likely, and only for a limited number of different species. Okay? We've heard quite a lot about phage, and there was a great news item. A novel phage therapy saves a patient from multi-drug-resistant Acinetobacter baumannii. They were going to die. There was nothing else could have saved them. Phage therapy saved them. Well, I actually believe phage therapy. The problem is nobody really understands the detail of the clinical studies where it happens in Eastern Europe. So you need to repeat good quality clinical studies in the main territories, Europe, Japan, North America. People have tended to try to use a traditional model. So that is, basically, they want to build a manufacturing plant that will make a phage product they can ship globally. And the same phage product gets used globally, and that way you make lots of money. The thing is, phage probably won't work like that. Phage will probably be a little cocktail of bacteriophage for this patient, or for the infection that's going around this particular hospital. And that will be different to one in a different country, or maybe a different part of the same country. Okay? And it's very difficult to work out how one big manufacturing plant can deal with the variation in product. So the future of phage, if it gets used, is probably going to be more like blood products, where you make a quality product locally, fit for purpose, okay, to guidelines, and it gets used. And that's more or less how it gets used in Eastern Europe. You'll have a microbiologist working across 3 local hospitals with a library of phage, works out which group is good for this particular patient, treats them. Okay? So it's not good for acute infections, it's probably good for those chronic infections where you can get the bacteria out and you can work out which phage cocktail to use. Very interesting that Amplify, one of the main phage companies, has announced a strategic emphasis on personalized therapies. So they've kind of realized that the global monolith with one therapy across the globe isn't going to work. Okay? So the A2A experiment, if you like, is that antibiotics have provided multiple medicines over 70 years, but there has been an extraordinarily large investment in human capital, resources, money, all working on it, and alternatives have just not had the same level of investment. And there's insufficient experience and literature of that transition from preclinical to clinical to understand what the risks and the benefits might be. The clinical potential is unproven, and all of this makes a very challenging investment argument. But we also recognise that there were new sources of medicine here. We just had to work out how to deliver them. So we've identified the approaches that may be most attractive, but to make use of those, we're going to have to develop diagnostics, and we heard from the last speaker the benefit. And if you have a specific, Pseudomonas-only agent, you better know that Pseudomonas is there before you use it. We may have to refocus healthcare from treatment to prophylaxis, because that's the way we're going with these therapies. We will need multiple products to replace a single antibiotic. So these new antibiotics over here will treat 20 odd different bacterial infections. Most of these products, the alternatives, will treat one bacterial infection, or even perhaps just a few strains of that infection. Funding into alternatives tends to be research push driven, right? What the academics are interested in. We really need to focus on market pull being an element of deciding which projects to fund. And we also have to bear in mind that we need to invest in experimental clinical medicine, not just drug discovery. Because if you just do clinical trials for drug discovery, the chances of you working out how to do this stuff are limited. If you realize it's going to take smart people doing experimental clinical medicine to understand the disease process and how these can be applied, you've got more chance of understanding how to use them. We have to develop the alternatives to antibiotic network and make a much more collegiate approach. One of the problems is the champions of one alternative approach don't really believe in the other alternative approaches. And so you tend to get research funding panels, and they'll say, well, we've got an alternative person on the board. Well, that's fine for their approach, but they're going to vote down all of the other approaches because they don't believe in them. And you probably need to be a bit more open-minded than that. We also have to accept that without adequate funding, we cannot act as if there will be replacements for antibiotics. And that was one of the big policy messages is, there's no use putting up phage could be inappropriate or antimicrobial peptides could be an appropriate replacement if we're not willing to invest in the amount of cash that we need to. So there is an opportunity, but there may not be enough activity. This is a list of the 19 approaches. We project planned out at least £1.5 billion worth of worthwhile activity, not to get these things to market, but to find out whether they work in patients at phase 2. There are many other approaches, some of the more traditional approaches that could be done as well. And so you can estimate that there's at least £10 billion worth of good research that can be done to support and underpin the future antibiotics, whether they're traditional or non-traditional. And £10 billion is a lot of money, okay? So one of the things the funders wanted to do was to achieve impact with this review and the publication. And many people will, of course, stop at the point of getting their paper published. But we didn't do that. What we did was we proactively marketed the publication. So I emailed 765 people in academia, industry and government, non-governmental organisations, charity and policymakers to make sure that they knew the paper had come out. Used my 1,200 connections in LinkedIn, used Facebook. My son set up a Twitter account for me and helped me work out what you do with that, although I'm not very wise, and kind of tried to keep the dialogue going and increase the impact and knowledge base. So that particular publication in just over a year has had 66 citations in academic literature. But more importantly, it's been used by policymakers. So I worked with the O'Neill Group and helped draft the Vaccine and Alternatives section of the O'Neill report. And in there we said alternatives to antibiotics should be treated on par with antibiotics. So we should be blind to the mechanism of action. If it works, it works. And alternatives should have access to the Global Innovation Fund in the same way that traditional antibiotics should. BARDA, the US Biodefense Agency that is very active in this area and which kind of incubated and created Carbex, which has got a huge budget for antibacterial development, went through the article and contacted every company that was mentioned to inquire about their programs and invite them into a dialogue with BARDA and Carbex. The World Health Organization has used it in their development of a pipeline to understand the future need. DNDi and the GUARD initiative in Europe used it as part of their portfolio analysis. Their aspiration is to create a fund to spend about 60 million euros a year on antibiotic development. I was invited to present to the Science Technologies Opportunity Assessment Group at the European Parliament to understand the future of antibiotic need, and the Pew Charitable Trust, which is similar to the Wellcome Trust in the States, in that it is a policy body informing the government over there. Elsevier every month look through the hundreds of journals and the thousands of articles that they publish every month and award the Atlas Award for Research Impact to one article. So they do 12 a year across the hundreds of thousands of articles that they publish every year, and they awarded, I think it was March last year, to this article, and it's still currently in the top 5% of all research outputs published by Elsevier. So it's definitely achieved some impact, which is what the funders wanted. So how do we solve the problem of antibiotic resistance? It's all about partnerships, because no one organization is big enough. And it's about bringing collective experience, skills, and insights together. And one of the reasons I created a working party that had dyed-in-the-wool, old-fashioned industrialists and this sort of new generation of alternative people together was to get those people talking, because they never meet each other, right? They don't talk. And one of the great things is that some of those sort of industry experienced people who have discovered traditional antibiotics have now taken scientific advisory board or non-exec positions with some of the alternative companies, so that you're getting that experience of clinical development and how to do this now, spreading into the alternative field. We have to work internationally because it's an international problem and no one country is big enough to take it on. It needs to be coordinated. And we've heard this in an earlier talk today. There's too much to do, so we shouldn't overlap, apart from in areas we agree to overlap in. And it's perfectly reasonable for one group to say, we're going to do this really well, you do that really well, and let's cover our bases. It needs to be sustained, and it needs to be sustained because we need to train people. You have to invest 10 to 15 years in somebody in this space before they can start to innovate and lead themselves. And we don't have infrastructure for that at the moment. It also has to be sustained because this is a generational challenge. There'll be people in 100 years or in 300 years having to deal with antibiotic resistance. Okay? And unless we create the infrastructure in place to put the foundations now, then those people are going to struggle. And we have the benefit of the 70 years of experience in traditional antibiotics. And there are people around now with 20 to 30 years experience in traditional antibiotics. But as we're not making traditional antibiotics anymore, there's probably 5% of the number of people working in antibiotics worldwide as there would have been 20 years ago. Soon, when those people retire and die, we won't have experience in antibiotics. So it's really important that we use that experience now to train the next cohort to go forward. And finally, we shouldn't be shy of talking about big science budgets, right? So I said 10 billion, and people in life sciences don't often see 10 billion and think it's okay. Well, look, the Large Hadron Collider costs $6 billion. The International Space Stations cost $96 billion, right? Antimicrobial resistance is probably somewhere between the two. But these things, they're groups of thousands of scientists and engineers working together to solve a problem, right? They're not trying to make money. There's no mini production line of Large Hadron Colliders coming along to sell to the public. Yeah? So if as a community we decide something's worth doing because it's for the good, right? You don't have to make money out of it. You just have to decide to do it. So I kind of think that in the future, a lot of the early discovery and maybe some of the early development needs to get done in large public-private type bodies like the Large Hadron Collider project. And then what you do, and these guys wouldn't be good at developing drugs and doing clinical trials. What you then do is you move it into pharma, and you just pay pharma on a cost-plus and a product basis to do it. Bearing in mind that if pharma haven't had to spend the billions and billions of dollars in research and development, they don't have to charge so much for the end product because they're taking less risk. All they're doing is doing the clinical development at the end. So I think that's an interesting model for the future. So I'll stop there and take questions. Thank you.
So that was a glimpse into what might be there for us in the future to use as sort of agents to treat infections. I talk a lot about ninja polymers, not knowing what I'm talking about. Is that the peptides or not?
Ninja polymers are kind of different. I mean, there's 4 or 5 of the flavors. It's a little polymers which, for charge or shape reasons, can punch holes in bacteria, and they are interesting. They're interesting for a number of reasons. Delivering agents. You can deliver DNA with them into cells. You can deliver antibiotics. Presumably, you can deliver antibodies. They could be used to punch holes and increase the sensitivity to other antibiotics. So again, it's just one of these things. You have to be open-minded. You have to do the pharmacology, the toxicology, and find out whether they have potential.
But they're not on your sort of
list. They weren't in the list. No.
I always tell the medical students that's the future because I like the name sort of thing. But
yeah, could be.
I'll stop talking about them. So, any questions, please?
Brilliant talk. It's actually allowed me to have a bit more faith that maybe something in the future. I was particularly impressed that they are actually not that expensive to bring to market, which is good news. My heart sank when I looked at the options available for gram-negative bacteria outside of Pseudomonas. So globally, we know Klebsiella pneumoniae probably have big worry. Any idea of see anyone focusing on that as a bug, a problem bug?
So a few of the companies have very early projects at the discovery phase. I'm not sure they would even be in preclinical models yet for Klebsiella. It's recognized as a problem. Most of the companies really not very well funded. They can't have deep pipelines, so they have to focus a product in the area of high unmet need that they can get money to do. But Klebsiella is on the radar for a few of the companies, but you don't hear about many other species.
Any questions from over here? Questions from over here?
Thanks very much. That was fascinating. Is anyone doing the same sort of work as you've done in the field of viral infections or fungal infections? They're big areas, and you focused on antibiotics.
Yeah, well, the viral space is much more mature in terms of novel compounds. It's easier to get money, and so there are biotechs operating and be able to fund themselves. Antifungals, you're right. We looked at whether we should do that, but then you'd almost need half as big a group again to get the right experts. I know in this country there's F2G, got a very interesting antifungal project. John Rex happens to be their chief medical officer. Antifungals are tough, obviously, because they're eukaryotic, and so the tox is more of an issue than going after an antibacterial. It's just down to medical need, and it's an area that probably needs more public-private support to get projects more mature until other people will pick them up.
Thank you.
If there's no more questions, then can we thank Lloyd? Thank you very much.